Okay, welcome everybody, welcome. I can see people already filling up the webinar. Please do come in, make yourself at home to our virtual event here at SimCell. I'm very, very delighted, very, very pleased to be welcoming you all here today. Please let me know where you're from, where you're calling in from in the chat or the Q&A, just so we can check that those functions work, and also just check that the audio is working well. Fantastic to see so many of you here. Let me just check the chat to see if we can see anyone writing in there, or check the Q&A. These are the tools that we're going to be using during the webinar to actually take questions from you and to ask our speaker, who I will introduce in just a second. So we've got Maine USA, hello, and welcome to you. And it looks like things are working very, very well. We've got a lot of people from the US, great. We've got Kentucky, New York. Fantastic. So we've got a lot of people from the US here, Tori. So okay, I think we can get started. This is exciting. It is my pleasure. My name is Ben Liberton from SimCell, and it's my pleasure to welcome for you all today Professor Tori Bengtsson from the Department of Molecular Biosciences, the Wenner-Gren Institute at Stockholm University. And we're especially excited to invite Professor Tori Bengtsson here today, because he's going to present a brand new method that has just been published in Communications Biology for the use of isothermal microcalorimetry to measure cellular heat production in bright adipocytes. Please feel free to ask questions in the chat during the webinar, and we'll put them to Professor Bengtsson at the end. But until pleasure to hand the floor over to you. Tori, welcome.
Thank you. Thank you so much. So again, my name is Tori Bengtsson. I'm a professor at Stockholm University and in physiology, and I've been working with brown fat for my whole life. So that is more than 25 years. And brown fat is a very nice tissue, because its main function is actually to produce heat. So I've had a dream for a very long time to actually be measuring heat production from cells, from brown fat cells and other types of heat producing cells. So there are a number of techniques out there that actually can measure heat, but all of them are measuring heat indirectly. For example, oxygen consumption, pH change, and so forth. But doing that is a little bit like being interested in lightning, and you have to suffice with actually the thunder. So I've always had this this dream of actually measuring heat directly. So about more than 10 years ago, I actually met Dan Hallén, who was the founder of SimCell. I met him at a conference at the City Hall here in Sweden, and I was telling him about this dream that I had, that I would actually like to measure heat from these kind of cells. And he said, wow, because I'm actually trying to build a machine that can measure heat from cells and bacteria. And then I got really excited, of course, because I thought, wow, my dream is actually to measure heat from heat-producing cells. So we decided there and then that we should do something together. And what we decided there was trying to understand, okay, how should that machine then work? And the machine has to be very, very sensitive, of course, if you're going to measure heat production from cells in suspension or on a plate. The machine needs to, of course, measure heat, and it needs to measure heat for a longer time period, so you can actually check the kinetics of it. And most importantly, you should be able to measure heat in different wells, so you can compare stimulation or not stimulated cells. At that point, Don said, yeah, I will build such a machine. And I said, yes, I will test such a machine. And he said, but I have a problem. I don't have any customer. So he said, can you be my first customer? And in a weak point there, I actually said, yes, I can be your first customer. So I paid him quite a substantial amount of money from my grants for a machine that was not there yet. So we didn't have any machine, but I had paid for a machine. So Don and others at Simcell started to actually build this machine. And I can tell you, all of you listening here, if it sounds easy to make a super sensitive machine that can measure heat from cells, it's not. It didn't work. At first, it didn't work at all, but some progression, we came more forward, and then tragedy struck. Don actually passed away, and I thought my dream was gone. But Simcell actually persevered. And I want to again thank Magnus Jansson, who has been working with building this machine for very many years. And he just struggled on with his colleagues. And in the end here, actually, Jesper Eriksson, who is the CEO of Simcell, came in, and they pushed this machine to the end and actually made a machine that was very, very sensitive. And I'm so happy to present the data we're actually getting from this machine now. So the machine works. And now I'm going to kind of go through a little bit of the different data we have from our point when we actually have this machine. Right. So why am I so interested in heat production? It's because of the obesity epidemic. And you all know that we are getting fatter and fatter. And I'm not going to show all of this going through the years. The point is that we are getting fatter, we're getting sicker because we're getting fatter. And anything that could actually stop this epidemic would be fantastic. So there are a number of adipocytes, 3 of them. The one which is most known is the white adipocytes. So that is, of course, the adipocytes that will take in fat, and you will have more fat in the cell, and you will have more fat cells if you grow obese. The main function of this is to store energy. But there are 2 other cells that are very, very interesting, and it's brown fat cells or brown adipocytes and bright adipocytes. The brown adipocytes have a lot of mitochondria in them, which makes them brown, and their main function is actually to produce heat. Then you have a cell type, which is something in between white fat and brown fat, which is called bright or beige. These cells also contain mitochondria, but not to the same amount as brown adipocytes. And they also have functions, and we believe that some of the functions is actually thermogenesis, so the production of heat. We are really interested in both of these cells, but here we have concentrated on the brights, because if you can show that you can stimulate brights and get an effect with this machine, it's very likely, and we have also shown that you can do that in brown adipocytes. So here we are actually focusing on brights. So the protein in these cells that actually produce the heat, or at least most of the heat, is a protein called uncoupling protein 1. The uncoupling protein 1 is situated in the mitochondria. And when you get protons going through the uncoupling protein 1, you actually produce heat. The interesting part here with this system, with both brown adipocytes and bright is that you can actually stimulate this heat production. So if you stimulate adrenergic receptors that are situated on these cells, you start a process that will increase the heat production through UCP1. You can do that either through your endogenous norepinephrine, or you can actually have other compounds that stimulate beta adrenergic or adrenergic receptors and stimulate heat production. So the idea is to find compounds that can actually increase and stimulate heat production. So do we have brown fat and bright fat in adult humans? For a long time, it was thought that we only had brown fat in babies, but nowadays, and we have shown and others, that we actually have brown fat in adult humans. And this is a picture where you can see that the brown fat here shown as black is induced in cold and cold acclimatization. This is actually not a measurement of heat production, but this is a measurement of glucose uptake, where you can radioactively label glucose uptake, and that is a way to measure activated brown fat. The take-home message here is that if you're in a thermoneutral zone, you don't have any activated brown fat. If you are in a cold situation, or if you are cold acclimated, you will activate your brown fat. You can also activate this brown fat by introducing a compound or a drug that would then do the same thing. And the idea is, of course, that if you activate this brown fat or bright cells, you will burn energy, and that will reduce the fat in your body. So you have the obese person, you will activate the thermogenesis, and then you will get slim and healthy. And that is, of course, another dream you can have. So to do that, to find these molecules, you need to have a system where you can actually test this. So you need to have cells, and you need to have a machine that can actually do it. And as I said, the machine needs to be very, very sensitive to actually be able to do this. And you need to have a method where you have your cells and your machine, and then you can measure this. So what kind of machine was actually then accomplished in the end? This is the Cal screener. The Cal screener is a machine that actually is able to measure very, very small changes in temperature. You can see here that this part here is actually where you put in your samples, and then you get your readings directly on your computer. So how does it work? First, you need to have somewhere to put your cells. And you have your plastic insert where you can put your cells. You can either put them in suspension, or you can actually grow them in the bottom of this well in different things, or you can put it in something that you grow your cells on, which you can put in. Then you take this plastic insert and you put it into a metallic container. And then you have to actually close that container with a lid. And then you have to put it in this plate, the Cal plate. And I can tell you that this sounds easy, or it looks easy, but it took a very, very long time for us from the beginning, and also for Simcell to actually understand how this should be done. Because if you just use the wrong O-ring here, you will actually get in trouble, because that in itself, the friction will actually produce heat. But that is solved now, so we're very happy with that. So what you do is that you take your samples in your cal plate, you put it in the starting position here with the machine, and then it needs to go into 2 different chambers. These first 2 different chambers is actually pre-equilibrating the temperature. So you need to get the temperature to the same temperature in all of the samples here to be able to actually measure this. Because if you go in with something there is a temperature gradient, it will actually destroy your reading. So this is one of the things that is very, very important with the cal screener, that you need to equilibrate this into these chambers. And then you need to push this into the chamber where you actually measure the heat. So what happens inside here in the chamber is that you have your vials, where you have your cell culture, your cells in, and I call this an open-close system. The open-close, which I mean by that is that it is open because you have air in there. So you have your cells, you can add, for example, 300 microliters of suspension of cells. And you can, of course, decide how much of the media you're putting in. And above that is air, so the cells will get air. But the system is closed. So at some point, the cells will actually run out of air, or there will be a pH change or something like that. What we know is that this actually takes very long time, and you can think about actually growing cells in normal incubator. It takes a very long time because before that actually happens. So the cells are viable for a long time. So when these cells are producing heat, it goes through a thermopile sensor and then into the heat sink. When it travels through the thermopile sensor, the heat travels through the thermopile sensor. This will actually be translated into a electronic signal that is then processed in the cal screener, and then you will get that out on your computer. So when we were doing this, as I said, we were very interested in bright cells. And bright cells you can get through different sources, but we decided to get these through mice and also from the inguinal part of the mice, where we know that there are lots of bright cells. So you need to dissect these out. And when you have dissected these out, you put that into media where you also put in collagenase and other things, so the cells should survive, but that should actually get separated from each other. Then you put this on ice, and then your mature cells will actually float. After a while, when these are floating, you take your mature cells and then you put that into the cal plate, firstly into the insert and then into the metallic vial, and then put it into the cal plate. And here it's very important to understand that you have some experimental wells and then you have some reference wells. The reference wells is actually used to get some of the background away, because you take the experimental wells and subtract the reference wells. Then you put it into the machine. As I said, you have to push it into the machine, and then each of these vials will actually go to their own thermopile and their own heat sink, which means that they are not influencing each other. So each of those are a single measure. You get a single measurement or a continuous measurement from these, and then you can look at this on the computer in real time. This process sounds easy and looks easy here. Or I don't know if it looks easy, but it was actually took a very long time to get this right. Now we have it right, and it was Carina Halleskog and Hamza Bukhari in our lab who made this happen, so we could actually get this working. And Hamza Bukhari is actually sitting here with me. So if there will be some very technical questions, I hope he will answer. He's a very talented PhD student. Right. So one of the things we understood from the beginning when we were working this is that the baseline needs to be very, very stable. And in the beginning, when we were doing this, we got big variation in the baseline, especially in the machines we had from the beginning. Right now, we have our machine in the lab is actually called Gandalf. It's because it's the oldest machine, and it has been resurrected so many times. Now it actually is very, very powerful machine, so it works, but it had some problems from the beginning. Now, if you look at the baseline here, it's super stable. You see that it's not much of a heat flow on any direction, and this is very good. So we're super happy with this, that the baseline is stable. This is a representative trace of the heat flow. So this is actually cells in there. And there are a couple of things I need to mention here. Firstly is that I said that you need to push this into these two chambers to equilibrate the temperature to get them to the same temperature. And then they go into the experimental chamber where we actually start to measure the heat. What happens is actually when you push this into the experimental chamber, you will have some frictional heat. So the first half an hour, you will have frictional heat, which is just generating by you pushing this plate in there. This frictional heat actually dissipates through the machine and will be away after half an hour. So after half an hour, there's no more frictional heat there. And then you get into a steady state where you actually can measure the output from the cells. So before half an hour, you shouldn't make any measurements because that's could be frictional heat. But after half an hour, and this we have actually gone through many iterations of, after half an hour, there is no frictional heat there. And we have proven that this is the case. So yeah, that's the frictional heat, and then you measure and you get the average heat flow from these cells. Another thing which is of course important is that the amount of cells actually should correlate to heat. So if you divide or dilute your cells, that should of course then be less heat. And we have shown this by doing different dilution series to see that the amount of cells actually correlate to heat. So what you see here is the heat output from the cells, and then we have diluted this, and this actually follows very nicely that the amount of cells actually correlate to the heat. So you can do a number of fancy mathematical measurements or afterwards. And here is what we were saying here is actually the same thing that you can do, measure the heat, and then you can do a dilution factor, and then you can see how these correlates to each other. And there is a very high correlation to the amount of cells. Why is this important? It's important because then it means that you can rely on measurements on this machine directly, and not due to kind of that there would be differences in the cell density, for example. So one of the things that we were thinking about was, okay, if we put this in this open closed system, are the cells actually viable? Are they happy in this system? So this is a measurement of oxygen consumption, looking at the cells to see if they are happy, and this is after 2 hours. We have done this at other time points, and we can see that after 2 hours, they are happy. They react to beta-3 agonist. So this is a beta-3 agonist that when you stimulate with this agonist, you should get higher increase in oxygen consumption, and we get that. So this is a proof of that the cells, if they have been in the cal screener for 2 hours, they are viable. If you look at this, you can see that they are viable after many, many more hours, but the function is going down somewhat when time is passing. And of course, that is due to the closed system. But we are really surprised actually that even after maybe 10 hours or even longer than that, you actually have quite viable cells. So what we are doing now is then, of course, so can we actually measure heat from brights? This is the experiment that shows that we can actually do it. So again, we took bright cells from mice. This is mature cells. They are in suspension. We put them in machine. We have decided to look at the first hour here after the half hour where you equilibrate the friction part. So you put it in the machine, wait a half an hour, and then we measure for 1 hour. You could go on for a longer time, but we think that is the most viable part to actually measure here. So what you see here, and this is actually the heat then from the basal cells. And then we treat these cells with CL316243, which is a specific beta-3 agonist, and it stimulates heat production. So, wow, yes, the machine works, and you can actually stimulate heat production from these bright cells. And it's actually a doubling of the heat. You can look at this as either as heat flow, or you can look at it as accumulated heat. And also in the accumulated heat, it's just another way to look at it. You can see that there is a remarkable increase of heat production. And you can decide upon a time point here, for example, after 60 minutes. And you can, of course, do it as a bar chart or anything else. And you can see that there is a big difference between the non-treated and the treated. And you can, of course, go in wherever you want with this, and you can keep it on for a very long time, measuring heat, which is fantastic. You can also do this, since there is a number of wells, you can actually do a concentration response curve where you do concentration side by side, different concentrations side by side, and then you can get concentration response curves for different, for example, drugs. So the machine works for the measuring heat production from brights. So what about this uncoupling protein 1? So as I said, uncoupling protein 1 is involved in the heat production, and this is known. So what we're doing here is actually using wild type mice. And again, you can see here that you have a basal, and then we treat with CL, and you get almost a doubling of the heat. I want to note that the scales here are different from the different runs. And this is because we are not doing any measurements of actual numbers here or protein or anything like that. What we're doing is, because we are relying on that, we are actually dividing the suspension equally into all of the different wells. And we can see that works, since we have done these measurements of the number of the cells that actually translates into heat. So what we do here is seeing that we get an increase in heat production. And again, CL does it here. If you do the same measurement, and now this is a new measurement in uncoupling brights from uncoupling protein 1 knockouts, you can see that there is not much of an increase. There is a little increase, which is then UCP1 independent activation of heat, but most of the activation is most likely through uncoupling protein 1 in the wild type. So our conclusion from this study is that uncoupling protein 1 stands for most of the heat generation, but there are uncoupling protein 1 independent thermogenesis in these cells. If it's a direct thing or not direct, we don't know. So to be able to utilize this for my dream, which is actually then being able to measure heat and also use this for screening for novel compounds, you also need to look at this for the receptor. So on the receptor level, are you able to stimulate different receptors, and are you able to take away these receptors and not get the response? So here we're doing this in wild type, and again we're using CL31643, which is beta-3 agonist, and you can see that there is an increase in heat production. The heat production with noradrenaline that actually activates all of the different adrenergic receptors is somewhat bigger, even though it's not statistically in this experiment. And I'm sure if we made this a couple of times more, we would get a statistical difference between the beta-3 and the noradrenaline. The point here is that you can stimulate the increase of heat by this beta-3 agonist. If you use a beta-3 knockout, there is no increase in heat production. So CL does, at this point, does not produce any heat production, showing that the effect is through the beta-3, the CL316243 effect is through the beta-3 receptor. The conclusion from here is that yes, beta-3 is very important in murine bites. However, if you use noradrenaline, which hits all of the different adrenergic receptors, you still get a response in the beta-3 knockout, which tells us that you can stimulate heat production via, for example, and we believe this is actually through beta-1 and some alpha-1 effects, but we can't test that. We haven't tested that here. So there are a number of ways you can stimulate bright thermogenesis. We believe it's beta-3 is very important, but other receptors are very important. And this is, of course, very important due to the late discussions in the field about which receptor is actually in humans, which adrenergic receptor is the most important in humans to stimulate thermogenesis. And there have been discussions about it if it's beta-1, beta-2, and beta-3. And this is, of course, could be tested in this system if you can find a very good model or cells from humans. So if any of you out there actually have some good brown fat or bright cells from humans that you think they could be tested here, give me an email, or if you don't want to collaborate with me, buy the machine and test it yourself. So we were also interested to see, can we do this in human cells? And especially could we actually measure heat production in adherent cells? So this is HMADs that are actually adherent. The other one is suspension. This is adherent cells. HMADs is kind of a model from or bright. It has they actually express some UCP, but it's not very high expression of UCP, but they express some UCP. So we were really interested. Would we be able to see an increase in heat production from these HMADs? Because that could also kind of show that the machine is very sensitive, and then that you could use a number of different cells to measure heat production. So the answer is yes, you can. If you look at we used a beta-3 agonist here, and you can see that you actually get some increase in heat production. It's not as big as in the murine or in the mice, but there's definitely a response, and the response is actually statistical. And you can look at, you can see that it actually is either through the heat flow or the accumulated heat or through the deciding on one place here. It's statistical and you get some effect. However, if you use norepinephrine, you actually get to quite very more robust increase in heat production, where you have a heat production about more than a doubling. This again maybe shows that beta-3 is not all that can stimulate heat production in human cells. And this is, of course, very interesting and something that we and others are really interested in looking forward more into. So I'm coming to the end of my presentation here. My dream from the beginning was to find a system, make a system where you can actually measure heat from these kind of cells. And I would like to find novel activators of heat production. We have actually done that. We have produced a couple of compounds that can actually increase heat production in these cells. And now we're back to mice and bright cells. And you can see that our compounds here, it's not reaching CEL, but it's actually reaching norepinephrine. So you have and it's dose dependent. So what I want to say with that is that you can use this machine for novel compounds and you can measure actual heat. And this was my dream, that you find something, have a sensitive method that you could actually do this in parallel. And this is actually then in parallel where you're running in one of these runs with a cal screener. So the conclusion is my dream actually came true. Actual heat is measured, which I think is fantastic, because I really like to look at the lightning and not the thunder. I'm not saying that you shouldn't look on the thunder and other things. I think this is also very important, but my dream came true that we can measure actual heat. You can measure adherent or floating cells. You can do UCP1 and UCP1 independent measurements. So if you're interested in these kind of things, you can do that if you have access to the knockout. You can use different cell models, human or murine, and I'm sure you can come up with many different ways to actually use this. You can actually see this in real time. You can do real-time measurements. You can look at the kinetics of the heat production, and you can actually look at this for long-term heat production, which I think is very important, because you want, if you're looking at, for example, after novel activators, you want something that keeps up the heat production. And you can screen for thermogenic activators. at the same time getting real concentration response curves at one run. I think this has been really nice and taken quite a long time to get it here, but I think in the end it actually came out very well, and I'm happy to have reached my dream in that respect. I'm open for questions.
Great. Thank you very much indeed, Tori, for the great presentation, and also the kind of journey through the development of the CalScreen. It's really nice to kind of see it all put together like that. So I appreciate that very much. Anyone, we've already got a ton of questions actually, but please, if you have any more questions, please ask them using the Q&A function on the webinar. If anyone has to leave but would like to learn more or contact us, then please do go ahead to our website, www.simcell.com, and you can find out more information or contact us if you would like any more information. So I think the first question we have is a technical question, which is, how long does it take to go through the equilibration chambers before the 30-minute wait for the frictional heat to dissipate?
The frictional heat and all, maybe I should actually get Hamza to respond to you. Hamza, do you want to respond?
Yeah. So it usually takes around 20 to 30 minutes for the frictional heat to die out, and we can actually check it through the reference wells. So when the reference wells have stabilized, we know that the system is in equilibrium, basically. So about 20 to 30 minutes.
Excellent.
Brilliant. I have a phenotypic question here. So does from the beginning of your presentation, does cold exposure actually increase hunger? So you actually kind of see this phenotypic change?
Yes, I mean, if you actually put mice in cold, they will eat more. So yes, you will eat some more, but usually if you increase thermogenesis, the increase in food intake will not be as much. So you will lose energy. So you will not actually eat. So you wouldn't compensate. That's maybe the question. Would you compensate? No, you wouldn't compensate fully. You will actually reduce your weight.
And related, I think, does cold exposure activate the brown fat, or does it generate new brown fat tissue?
So cold exposure both activates brown fat and it also generates more brown fat. And this is where maybe the conundrum and a big problem when you're actually making a drug for this. The dream there would actually be to have something that could activate brown fat and also make more brown fat, which is not as easy said as done. So yes, you normally in cold you do both. You stimulate the effect of it and you stimulate the amount. Great.
And we have another question here. Is it possible to measure heat production in white adipocytes or 3T3L1 cells?
Yeah, so I mean, it's possible to measure heat production. Heat in basal state, you can measure any cell, I would imagine, in this system. The question is if you can actually stimulate heat production. And I think you can to some extent, but not to the same extent as brights or brown fat, because that they have their own generation of heat production. So if you poke the cell with some different agonists, for example, yeah, you will probably have a higher increase in metabolism, and that could actually, you could possibly measure that. We haven't done that, but what we are interested in is the stimulated, where this actual great stimulation of heat production. But yes, you could. That's maybe the short answer.
Does the cell cycle influence the production of heat, and how do you synchronize cells in that sense?
We haven't synchronized the cells in any way. I think possibly you could look at this. We haven't done it. I don't think it actually would be very big differences, but that's my feeling.
I have another question about the frictional heat, like the nature of it. Is it the same like in every experiment? Is it the same across all cells, or is there any variation with the experimental setup?
Do you want to answer that, Frank?
So we have seen some variation in terms of the maximal frictional peak output, and we have gotten many questions over the years that is it actually affecting the readings that we take later? So we have done like a lot of correlations to actually see if the maximal peak frictional output is actually influencing the data that we collect. And so far, we don't have any indication that it does.
Great. Have some questions about the bright data that you showed. So in time zero on the experimental slides, is this the time after the equilibration, so after the 30 minutes? How do you set the time zero?
I didn't get that. Once more, can you? Is it time zero in the experiment?
Yeah, in the experiment, how do you set time zero? Because you've got this equilibration phase for kind of 30 minutes.
So we set the time zero according to the heat exchange that is going on between the reference wells and the thermopile. So once that reading has stabilized, which takes around 20 to 30 minutes, we know that's where the time zero is, basically. Like when the entire system is in equilibrium.
Great.
And we have tried to keep it consistent for all the experiments. Like it's usually 30 minutes and we start collecting the data afterwards, making sure that the reference wells are stabilized during that time.
Great. And in the bright adipocyte experiment you did, what was the cell concentration that you used?
So we didn't actually measure the cell concentration here. We're actually doing what we're doing is that we're dissecting, we let them, as we said, that you do the digestion, so they separate, and then we take a certain amount and then put it in. We're not adjusting for cell number in these experiments. This is something you could do, but it's very cumbersome and takes a lot of effort for you to actually get this right. It sounds easy, but it's not as easy as it sounds. So this is something you could do if you are really interested. We are more interested in that we actually get the same amount of cells in all of the plates, and that is actually depending on the volume. So we divide it on volume.
Okay, great. Can we add chemicals to the plate during measurements like the seahorse system?
No, I mean, this is one of the I wouldn't say downside, but it is something that would be nice if you could, but since it's a open closed system, you can't. I mean, when you put your cells in there, that's it. Then you have to kind of do your measurement as is. So you can't put anything in there while you're doing the measurement. Yeah.
But I guess we kind of say when we compare ourselves there, you know, that you're actually measuring direct heat. So that's the kind of benefit of the cal screener and using microcalorimetry in this kind of experiment, right?
Yeah, I think it always there's some discussion which of these techniques are the best. And I don't really dislike that discussions because they complement each other very well. And if you want to actually look at heat, there is nothing out there really that is similar like this. So I think everyone should just look at it like, okay, what kind of extra benefit can you get from these things? And that's the discussion, not which one is best, because there is no best in that respect. Different ways to look at it.
Okay, so besides oxygen availability, how does the media composition affect heat production? So for the example given here is, did you test galactose media, for example? Do you have any idea what the difference is?
No, we didn't test galactose. And I'm sure Martin Schieber is right there that this will actually affect. We don't think that effect is very rapid. It was probably be after very many hours in the media. And of course, this could actually affect the output. And this is something that would be interesting to look at, but we haven't done that yet.
Yeah. Question about detectable heat from a brown adipocyte in a monolayer culture. So can you actually get detectable heat from a monolayer of brown adipocytes?
You want to take that?
So right now we haven't tried it, but we're currently in the process of actually doing it, comparing different differentiation mediums and correlating the amount of UCP1 to the stimulated heat production. But I can't give you like a hard answer right now because I haven't done it. But our data with HMAT seems to suggest that it should be possible, because as compared to, let's say, brown adipocytes, HMATs do have a lower UCP1 amount, and we can still detect stimulated heat production. So I think even in murine brown adipocytes grown in a monolayer, it should be possible.
Yes, that's my feeling also.
Cool. Are you planning to look at mechanisms in obesity-related diseases?
Yeah, I mean, we haven't thought about it in that way, no, but maybe that's the question. That's the answer.
Send us the cells.
Yeah, exactly.
Nice. So we have a question here, a technical question about, so the data for the heat flow and accumulated heat show differences in the levels between the wild type and the knockouts. So like the UCP1 and the B3 knockouts in different experiments. So they're wondering if there's been any adjustment made. I guess it's not been made to cell number, but did you adjust them based on the volume or anything like that?
So for all the isolations that we've done, like for every genotype, basically we keep the number of depots that we're isolating cells from the same. But what we have noticed is, like, according to, and the protocol is done in parallel, especially if we're comparing genotypes. But what we've actually noticed is that the basal level of heat production from these different genotypes can be very different. And we haven't really corrected for the differences in basal, but which is why we have presented the data as fold change as well, because you are essentially comparing each experiment to its own baseline level. But we haven't done like any other adjustment beyond that. Like what you see on the accumulated heat and also the heat flow is basically the raw data that we get from the machine.
Yeah, I mean, you could do a lot of different ways to actually do this for protein, for cell number, but we actually decided to actually show the kind of the raw data fully out there. And my feeling is also like what we're more, as I said, more interested in is that it actually is the same amount in each well from each run, and that we're pretty sure that it actually is. So there are the same amount. But comparing different runs is much more difficult. And I think that is the next step in actually getting this method a bit further to get that in some way. And it needs to be not too cumbersome, because then it's very difficult and it takes a long time. So it needs to be something that's quite rapid. We have been thinking about it and we have some ideas, but yeah.
Great. And if you measure for days, can you differentiate between heat production and cell proliferation?
If I mean, what the question is actually, proliferation will generate some heat, and I don't know if we can say that we can. We don't know. We haven't tested that. It's actually quite a clever question. I mean, I'm pretty sure you can actually measure proliferation here. That's not the question. Differentiation is, of course, much more difficult. That's not what we have been kind of interested in. We are actually looking at the stimulated thing. And of course, the stimulation on, if it was proliferation, there could be some change there, or that you increase the proliferation. It's actually a clever question. We believe in the cells we're looking at here, the brights, the mature ones, that's not an issue. But this is something you could actually look at. Yes.
Interesting. Did you ever manage to compare the same cell model in both an adherent and the suspension to see if there was any differences in the heat production?
No.
Nope, we have not.
No. A quick answer, no.
Do you add your thermogenic activators before the cells enter the system, or can you add them during the experiment? I think we've kind of covered that, right?
No, you have to do it before, and of course that's where other techniques are more rapid than this technique. So then you can look at minutes, but I think the upside here is actually that you can look at long-term changes in thermogenesis and so on. So no, you can't with this technique.
So we've got a question about resolution of the sensor here, what you're actually measuring. Are you measuring kind of heat production on the cellular level, on the subcellular level? Can you elaborate on that a little bit?
I think, I mean, you can't know where the heat comes from. It comes from of course, the subcellular level. It's mitochondria. Then it dissipates through the cell. So it's not possible to know where it comes from, the heat here. It comes from everywhere. All the chemical processes that are happening, and this is why I think this is so interesting. This method, that it's like the accumulation of everything that is happening, and that is why I also want to have that dream to actually measure heat, because then you catch everything that is happening. So no, you can't see the subcellular. I mean, what maybe you could think about if you could use mitochondria in there and measure mitochondria. We have thought about it. We haven't done it yet, but it's one of the experiments I would favor.
Fantastic. Now I see the clock running, and we're kind of getting close to the end. There's basically zero chance of us getting through all of these questions before we reach the hour. So I was wondering if you had time to stay on for 10 minutes to go through them, or we could maybe find another way to answer the questions, to provide answers to all of these people. What would you prefer?
I could stay on for 10 minutes, but I also, but we had some.
We have something after.
We have something after. I can't stay on for 10 minutes. Nope.
Okay. So what I'll say then.
I'll address the questions through like private message.
Yeah. I mean, I think we could, if someone would actually want to send this, preferably to Hamza.
Well, we have recorded all of the questions, and so we'll make sure that we get answers to everyone. So if you've signed up to this event, we will email you afterwards. We've recorded, and we'll send you the recording, but we'll also try and get some of the questions answered for you. So with that, I'd basically just like to thank everyone who attended and listened and asked your very, very insightful questions. But I'd also like to really thank you, Tore and Hamza, for spending your time to really, to develop this amazing method and this amazing use for an instrument that we're very, very proud of, but also for coming here today and telling us about your experiments and the development. So thank you very, very much indeed.
Thank you. Thank you so much.
Thank you.
Thank you.